I love our intro more than I love anything else, honestly.
Oh, that music rocks. I could dance all day.
Okay, except you wouldn't dance all day. You wouldn't even line dance when we went to the saloon, Professor Jordy.
I line danced for about 30 seconds, okay? I did 30 seconds of line dancing.
30 seconds is never enough.
Welcome to Absolute Genius, a new podcast series from Thermo Scientific. I'm Jordan Ruggieri.
And I'm Cassie McCreary, and our genius for today's episode is Sarah Philo, a recent PhD graduate from the University of Washington.
Sarah specializes in wastewater surveillance and epidemiology and has used her work to learn about SARS-CoV-2 and how it travels through communities. In this episode, we talk about PCR, life as a PhD student, and a little bit about feces. Actually, a lot about feces.
A lot about feces. Heavy on the feces. I love it.
Well, Sarah, thank you so much for joining us on Absolute Genius. It's an absolute privilege to have you here. We're really excited. I know, we've talked offline and dove into the amazing world of wastewater epidemiology and talking about especially SARS-CoV-2 surveillance in wastewater. At this point, a lot of people have definitely heard about the SARS-CoV-2 virus. But I feel like there's still some room to learn about what wastewater surveillance is, what that looks like. Can you give us, a little bit of background? What do you do? What does that world look like?
So I actually just finished my PhD in environmental health at University of Washington, Seattle. My dissertation was a lot about wastewater surveillance for SARS-CoV-2 and then also some antimicrobial resistance. What the world looks like, that is a huge, huge question. I mean, people are historically have been looking for things like poliovirus in wastewater for decades. Now it's sort of transitioning into a lot of different infectious diseases. People have been looking for like drug residues for a while. So it's just, there's a lot of different things that you can do with wastewater and what you can look for.
Describe that process a little bit. So wastewater, it's a collection of sewage, and what does that actually, what does that look like?
Do you want to ask what that looks like?
Maybe I won't say exactly what it looks like. That might not be appropriate for soft stomachs. There's a couple of different places that you can collect samples. So one thing that was implemented a lot during the pandemic was actually collecting either at like dorm effluent on college campuses, so the pipe that comes right out of a dorm, or from like a manhole on campus that collected from a few dorms. And that would give you an idea of like a smaller community. And that was for colleges that were doing a lot of like regular testing of their students, and then they could actually isolate students who were sick. You can also collect at like the effluent from hospitals. So if you're looking for what's going on in that hospital, a lot of the work that I did in my dissertation was actually collecting at the beginning of the wastewater treatment plant, or the influent as we call it. And that gave us an idea of like the entire wastewater catchment area, everything that is collected by that treatment plant.
I hardly know anything about this, so this might be a very ignorant question, but you guys literally, you just, like, somebody goes in there and they scoop a sample and there it is?
So a lot of my samples were literally just like somebody took an Nalgene bottle, like, the liter size water bottles, dip a bucket, and then you fill up that bottle. And that's called like a grab sample, where it's just like a one-time, you just grab it, you go. There are also things called auto samplers, which like it's a machine that you hook up at the pipe level. Every 15 minutes, it will collect some of the wastewater. And you can do that over the course of 24 hours, 12 hours. So it will give you like a composite sample of like all of the wastewater over the course of 24 hours, usually is what those are.
Automated definitely sounds a little bit cleaner.
Yes, also much more expensive.
We've got expensive taste. What can we say?
So what happens after you get that sample? Let's say you've detected some levels of SARS-CoV-2 in the wastewater. Has your research led to maybe some policy implementation, or how does it help guide what's going on out there?
I wish I could say that my research directly guided policy. That's what we hope for. But my work definitely focused more on the like research side. So we were never necessarily collecting samples to guide public health. It all sort of comes back to this like use case question of like, what is the goal? Early on in the pandemic, there was a lot of lofty goals of thinking like, oh, we can estimate the number of people who are sick. But that's pretty difficult depending on which catchment area you're looking at. So a lot of the work now has transitioned to looking at trends. So is SARS-CoV-2 rising or falling, or is it staying stable? And then also looking for those variants. And then there's a lot of push now also to look at like, presence absence in long-term care facilities. So like if you have a retirement home, you could test the wastewater there. It really sort of depends on like what the goal is of what actually happens with that data.
Yeah, that makes sense. Diving into, looking at maybe how many people were sick. What was kind of that indicator, or was there an indicator that something like that might be doable? Is that something that's still actively being looked at as a way to kind of translate that, those different trends that you're seeing? Or is that something that's mostly, it's mostly more on the surveillance end and not really looking at predicting population outbreaks?
Some people are doing it. I think in certain water catchment areas, it would be more feasible. My context for a lot of this is Seattle, which gets a lot of rain in the winter. And the Seattle wastewater is a combined sewer system, which means that we get road runoff as well as the sewers. So in the winter, when we saw a lot of COVID cases, we're also getting a lot of rainwater. So I think in some places it is much more feasible potentially. And it is something that's still being looked at. But the big problem with being able to estimate actually how many people are sick is there isn't necessarily the best like SARS-CoV-2 fecal shedding data. So that's like how much SARS-CoV-2 virus is actually in feces. And there was some good data from like early in the pandemic, but we're on variant 5 or 6. So how do those variants affect fecal shedding? And once that data gets filled in and improved upon, there is the potential to actually understand more about how many people might be infected. But that's a huge question right now, is this like fecal shedding piece of like how much virus are people actually releasing into the systems.
And you mentioned about rainwater and kind of the runoff there. Does that impact any of your results, or does that dilute out the virus and make it harder to detect or quantify?
Yeah, so the thing with the Seattle area wastewater is these treatment plants on average process like 70 to 80 million gallons of wastewater a day. So then when you get flow of rain on top of that, you can get over 100 million gallons a day. Even at the peak of the pandemic, there's a few thousand people, 10,000 people who are sick at once. Like, the world is huge and there are so many different situations. I can't really generalize, but more with like our data, it was definitely, we were able to detect SARS-CoV-2 pretty consistently. That quantification piece was a little tough because we were frequently detecting it basically like at our limit of detection, where it's like, okay, we know it's here. We just have no confidence in like being able to put a number of virus copies on this number. We could have done a lot of estimating of like, okay, if we applied this number to what we could have estimated if we had sampled the whole thing, but then you're just getting like so much uncertainty that we were more focused on like, is it here? Is it not here? Rather than putting like hard numbers on it. But again, that's just a function of like how it worked for us.
I didn't actually think about, when you're talking huge volumes like that, how much that virus itself could get diluted, I guess just depending on what's going on in the population, right? So, if you have a smaller outbreak, it's even harder than if you get even more people. How do you actually go about detecting the virus? What did the methods look like there?
So there were also, people were doing various forms of digital PCR, which tends to be less affected by environmental inhibitors. We decided to keep with the qPCR just because it's what we had done the whole time, and then we could have like this big cohesive data set, knowing that it probably wasn't as sensitive as something like a digital PCR would be. Some people are trying to work with different like rapid PCR protocols. So there's a lot of stuff that has been developed and is being developed right now for SARS-CoV-2 and wastewater specifically. But most of the work, I would say, is qPCR and then various forms of digital PCR. And then there's a lot of sequencing that goes on top of that to figure out what variants might be there.
And I can imagine just thinking about, a nasal swab, and you get a lot of viral load from that particular nasal swab, and then comparing that to the millions of gallons of wastewater, you can kind of puts in perspective the challenges that come with SARS-CoV-2 surveillance in wastewater. It makes it a lot more apparent on how hard it is just to detect and then utilize and quantify that particular data. Kind of going into the methods a little bit more, you mentioned differences of qPCR and digital PCR. Did you utilize digital PCR, and what did that look like for your research?
We didn't get a digital PCR system until about halfway through some of the work. So a lot of the digital work that I did with SARS was involved with comparing detection between PCR and dPCR. And then we did, whenever we would get a new positive control, we would quantify that on the digital PCR just so that we knew what it was like, instead of just going by whatever the lot number was that they gave us, just to make sure that those numbers lined up and everything. But we, again, just for consistency, I didn't do a lot of the SARS work on digital PCR. So most of that is just qPCR data.
Based on your knowledge or what else is going on in your lab, are there other ways you're utilizing digital PCR in terms of SARS-CoV-2 surveillance or in other organisms? Are you looking at possible other organisms to take a look at and try to bring up surveillance programs for those?
That's the big question right now with wastewater surveillance, is how do we take all of this expanded capacity and interest beyond to the next thing, right? SARS-CoV-2 is basically everywhere at this point. But how can we use a lot of this to look for other things? And one of the big pushes has been to look for antimicrobial resistance in bacteria. That has been a problem for medicine for a while and is only going to become a bigger problem. I did another piece of my dissertation actually looking at antimicrobial resistance. One piece was looking at if those resistance genes increased along with the first late 2020 COVID peak. Doctors prescribed antibiotics because they didn't have a lot else at that point for COVID. And for people who were hospitalized, it was to stop them from getting secondary infections. So we were asking, do any resistance genes increase along with that time period? And then I also did a little bit of a comparison between digital PCR and qPCR.
What did some of your results show when you actually were to compare those methods?
Like I mentioned, we tested for some different antimicrobial resistance genes with qPCR and dPCR. And pretty much all of them had a higher quantification with dPCR than with qPCR, which makes sense, at least knowing all of the inhibitors in wastewater and environmental samples tend to make qPCR a little bit less efficient. The one thing that I would say for anyone working with environmental samples is that I found it was really helpful to test them in qPCR first to kind of get a general idea of how much was there. Because with digital PCR, each reaction is more expensive. And if it's very easy to overload the compartments or diluted pass beyond what can be detected. So just kind of having an idea of roughly how much is in your samples before, whether you do 1 every 10 or whatever. So I found that was really helpful, actually, knowing okay, I think there's roughly this many gene copies in this sample. I'm going to dilute it out this much so that it should be within that band of optimizing the digital PCR.
You were able to actually, determine maybe a little bit more precisely the gene copies that were there using digital PCR over qPCR after kind of looking at that optimization kind of between the two?
Yeah, so I didn't do a lot of variability testing, I guess, of what is the standard deviation around these. I will say that the gene copies that we detected in digital PCR were higher than what we detected with qPCR. So this is a big problem with wastewater. It's just when you extract everything, a lot of times the inhibitors are also still present, just because there's a lot of things in wastewater that can be kind of hard to clean up. It's pretty well established that like PCR and qPCR do have some effects from those inhibitors, but with digital PCR, it tends to be less of an issue. So, like I said, I didn't do any like variance work of how much with a not variant with a SARS-CoV-2 variant, but variants with a C. Are we more confident? Like, what is the confidence intervals on these? But I did see that the numbers were higher with digital PCR.
Yeah, it makes a lot of sense. Where do you see the future of wastewater epidemiology when it comes to digital PCR and implementing digital PCR as a method for different surveillance programs?
Wastewater surveillance is talking about its future and like where it's trying to go. But I think digital PCR has a really good place at having higher efficiency of those reactions. So a better idea of like what is actually here and how many numbers are here compared to qPCR. And I think it could really help with finding some of those more rare targets on like a presence absence basis, because if something in your wastewater is at the limit of detection of your qPCR assay and then you throw in all of that inhibition on top of it, it's just going to be a lot harder to find. Whereas if with digital PCR, if you get rid of some of those inhibition effects, you're probably more likely to find a lot of those rarer targets than you would be otherwise.
Taking a quick break from our conversation to tell you about Applied Biosystems Quant Studio Absolute Q dPCR system. This instrument enables quantification of your targets without the need for standard curves in only 90 minutes. Digital PCR can be as simple as preparing your samples, loading onto the plate, and running the instrument.
Unlike other digital PCR systems, the Absolute Q dPCR instrument does not use emulsion or other droplet-based methods to compartmentalize reactions. In fact, the microfluidic array plate technology enables consistent delivery of more than 20,000 microchambers. It's a great solution for anyone looking to quantify gene codes.
And Thermo Fisher Scientific has a suite of dPCR assays for applications like AAV viral titer quantification, liquid biopsy analysis, and wastewater surveillance. You can learn more at www.thermofisher.com/absoluteq or visit the Absolute Genius webpage. Again, that's www.thermofisher.com/absoluteq or visit the Absolute Genius webpage.
The Applied Biosystems Quant Studio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures. Let's get back to our conversation.
I think for a lot of the people who are maybe listening who are fresher in their careers and still maybe deciding what paths they want to go down, or even people who are looking towards grad school or even that type of thing, has your career gone the route that you would have expected? Have you always been interested in sort of maybe, I guess, like a public health lens in terms of that? Have you always been interested in science? I know, like for me, even still today, sometimes I don't know what I want to be when I grow up.
Oh gosh. So I've been interested in science forever. Like as long as I can remember, I always thought science was like my favorite class. And then I did my undergrad in biology just because my university didn't have like 45 science degrees. And I was like, I like natural sciences and life sciences. So I was like, okay, I'll do biology. And then I was able to do a track in infectious diseases. And I was considering going into medicine, but thought I would get frustrated with like how many medical problems are preventable. There are so many things that it's like, oh, well, you wouldn't have this problem if you didn't grow up next to a highway and now you have asthma because you inhaled car fumes all day. So then I was like, okay, maybe let's try public health. So then I did a master's in global health, and my master's research project focused on zoonotic diseases, which is when infectious diseases jump from animals to people. And I ended up really liking that. And my master's advisor, he worked a lot in One Health, which is this idea that animal, human, and environmental health are like completely intertwined, and you can't study one or fix one without thinking about the other pieces. And one thing I found with that was that the environmental piece was kind of always missing. People tended to study animal health and then also human health with the people who worked with those animals, but they didn't always think about this environment piece. So then I decided to do a PhD in environmental health to better understand some of that. If you had asked me in high school or in undergrad if I would be here studying wastewater, I wouldn't believe you. It was just more little pieces and then little suggestions by professors along the way who were like, oh, wow, you're talking about this thing. Have you considered looking at this or this piece? So just like small conversations with, different people along the way. But I would not have thought that I would have ended up here, especially let alone with a PhD. I don't even think I knew what a PhD was in high school.
If you were to go back, or if you were to give somebody who's either just starting their career or just moving towards grad school, or if you could go back and tell yourself any piece of advice along the way, what would you say?
One thing I would say is everyone around you has this idea of what their path is. It can be really stressful of oh my gosh, everyone I know wants to do this thing and they know that they want to do this thing. And a lot of the time they're just saying that because they feel like they have to. Most people kind of just figure stuff out, and not to be stressed when you don't always know, because a lot of people don't know. And just to be open to new opportunities and to have questions and to seek out people who are willing to answer those questions. I really got lucky by talking with people along the way of I don't know if I want to do this, but I'm interested in these things. And it was just people suggesting things to me of oh, I think you would be a good fit for this class or this program. And yeah, just, being open to everything. And there are classes that I wish I had paid attention more in undergrad, because I was oh, this isn't what I'm interested in. And now I'm oh, I really wish that I had spent more time understanding that, because now it would be relevant. So just taking advantage of all the opportunities.
Yeah, it's excellent advice. And I think you hit on something really important, which is kind of that stress and being surrounded by people who seemingly know exactly what they want and things like that, and seemingly know exactly, what to do all the time and things like that, which leads me to my next question, because we love a segue. Do you have any oops lab moments or funny lab moments? Because I think it might be very comforting to our listeners to hear maybe about some of those moments where you're oh, maybe I shouldn't have done this.
Oh, I mean, there's always the oops, I autoclaved the wrong type of thing, and now our autoclave is a puddle of plastic.
Oh,
so that is always, everyone who's worked with an autoclave, I think, has that as an example. Yeah, we melted something once in the autoclave, and it was one of the huge, giant, 10-liter bottles, I think. And
oh, no.
A huge, huge plastic bottle. And we ended up putting it on the wall with the sign that says feces happens. Just, sort of a pun.
I want a t-shirt that says that.
I know.
I love that. I love that.
I've gotten to the end of a PCR run and then been oh, I didn't add the primer. Oh, that's 3 hours of my life. Just small mistakes, big mistakes, accidental oh, sewage spilled on the counter, gotta stop everything and clean that up. Yeah, so it's nothing that was ever catastrophic, I don't think.
I just wanted to also ask your best lab moment, a big contrast to that. I mean, what was probably maybe your proudest moment, or just something happened and you were God, yes, finally.
I have two examples. So the first is early on in my PhD, we were trying to insert a plasmid into a bacteria, and I had been working for a year on this project, could not get the gene to go in, and then finally it worked. And it was just I don't know what happened. I don't know what I did differently. That was just exciting because it was something that I had been working on for a really long time. But honestly, I think the best thing about my lab work is just the people. We spend hours and hours together, and we like it because of the people that we get to work with. There's this popular image of a scientist slaving away alone for long hours in a lab, when it's you're never alone. There are always people who are there with you and really make it worth it. Like I have a PhD. I did a PhD project, but I would not have gotten there without dozens of people in the lab and around the lab who helped and facilitated and supported all of that.
And a great team can make all the difference in a lot of ways. So yeah, that's that's awesome.
Yeah, you're not alone in the lab cave, right?
No, no.
I actually want to go back to one thing. You just completed a PhD. What was that process like? Can you give some insight into that process? What did you think of it? Anything you struggled with or loved doing as part of that?
I liked it, which I know is not common for a lot of people who get to the end of a PhD. Yeah, like I said, the people made it worth it. It was a lot of work. Each school has its own very different PhD process. They all kind of have like similar structures where, sometime in the first year or second year, you take like a qualifying exam. So that's kind of that one step to make sure that you like know that you want to keep going and are prepared to keep going. And then within the next like year or so, we call it a general exam. It generally involves like writing your project proposal for the PhD. And then you defend that proposal to your PhD committee, which is faculty in the department and at the university. And then at the end is when you do the like dissertation defense, which is you've collected your data, you've analyzed it, you've written about it, and now you're going to present it to your peers and faculty in your committee and defend your work and why you did what you did. And along that process, like your committee and your PhD advisor and your research group members, like they're all helping you along the way. So usually by the time you get to that dissertation defense, it's kind of like, oh, you're going to pass because they've all approved everything up until this point. It's just like, oh, can you analyze the data this way, like instead of the way you did it before? Like small changes, but by the end, hopefully the dissertation is like mostly complete.
That would be the most nerve-wracking, I think, for me, is standing up there and trying to defend your data. And you get this, impression of this mean group of people that just picks apart everything and throws things at you while you're up there on stage. But it doesn't sound like that's really what it's like.
Yeah. Well, luckily I was able to pick my own committee. So I was able to pick people who I knew would not try and pin me into a corner with something I don't know, which is something to think about too when you're picking, if you are choosing to go into grad school, the culture around that. Like, is your department going to be more supportive and more collegial and like making sure that you learn, or are they trying to make you defend your knowledge? And like, so that's a culture thing. Like by the time you get to the dissertation, it's much more of a like celebration of like, here's my 45-minute to an hour presentation of everything I did, and like celebrating all of that while also making sure that you like know what you did and like can talk about what you did intelligently. But again, like it could be totally different. I've only done this once. I don't plan on doing it again. So maybe different places have different options of like how they do that. But that's a big cultural question if people are looking at grad school. Like, how does this process go? Like, are they more collegial, or do they tend to be a lot stricter and harder on the students?
I mean, according to Jordan, the way he was describing what he was picturing, it sounds like it's like a medieval Times Square.
I have heard some of the like European systems I've heard tend to be a little bit more like that, where they are like grilling you on your knowledge and you have to answer correctly, and it's like 4 or 5 hours long. My experience, and most people I have talked to, it tends to be the dissertation is more of a celebration because all of the hard work went into it before then.
And this is totally me speaking with no PhD at all, so whatever. But I just feel like, as an outsider listening to a lot of this, I think it's probably pretty important to, like, there's a good degree that you're passionate, not just about science or whatever, PhD you're going for, but like your topic. And I mean, you've got to have that passion there. What is it about either your topic or just like science as a whole or whatever that like excites you, that makes you passionate about it? I mean, I always argue that science is like the coolest. It's like the punk rock of academic like disciplines that you can study. It is the definition of screw around and find out. It's the backbone of it is curiosity. It's the coolest.
I've never heard science described as punk rock, but I might start using that. The passion is like super important. And that's one thing I tell people, like, if they're deciding to do a PhD and they're kind of on the fence about it, I'm like, you can't be on the fence. You have to want it, and you have to know why you want it, because It's a lot of time of your life with very, very low pay. I'm lucky that I was funded the whole time, but like some people have to scramble a lot more to find money. And even then it's like you're getting paid for 20 hours a week when you work a lot more than that. And you have to remember like why you did it. In terms of like why I picked public health and sort of what the passion is, I really think it was just seeing so many problems that we could solve better if we just worked at prevention instead of treatment, especially with environmental health. There are a lot of things that we just don't have necessarily the best data on. So it's like, okay, we know this is a problem, but we don't have a good idea of like the scope of the problem. Wastewater surveillance can hopefully help shed some light on a lot of those pieces. And it was really easy in the pandemic too, to be like, this is important. This is a new virus that is all over the world. How can we better understand where this virus is, how much of it's a problem, and just knowing that like you can produce the data that, you know, public health might use one day. Yeah, the pandemic was kind of a little, a PhD in a pandemic is not something I would recommend to anyone, but it made it easy to know why, right? Like everyone cared, and suddenly everyone cared about wastewater. Because everyone was like, oh, I saw that thing on the news about wastewater. And it was like, yeah, I know, this is what I've been saying for years. And now everybody suddenly cares. Like, so that helped a lot as well.
I mean, we touched on this a little bit when Jordan was talking to you about like the process and everything, but grad school as a whole at the different levels and everything else, like it's a grueling process. And even if you are really passionate, I would wager at one point or another, everybody kind of like wavers, so like what's your advice maybe to somebody that's like questioning, well, do I want to move forward with this? This is a lot, like whatever. I mean, is it just to remember your why? Is it to figure out exactly what your why is? Like, what would you suggest?
Yeah, I mean, everyone wavers, right? Like it's exhausting. You come home sometimes and you're like, what am I doing? Like, why am I? And you have to keep that like why piece there. Why did I want to do this in the first place? And you have to like remember that of like, I loved the research piece. I was really interested in understanding a lot of this and, you know, pushing the field forward. Because if you don't have a good why and if it's just, oh, I'm going to grad school because I don't know what to do next, like that's not going to get you through. There has to be like, you have to want a specific outcome. So with like a master's degree, that could be more like a specific skill building of like, I want to learn these skills in this project, or like a master's in public health. Like I want to do more public health practice type thing. So that's why I'm doing this. Whereas with a PhD, it is a research degree. So it has to be like, I am doing this for the research because I want to develop those skills and push the field forward, and whatever that field is.
That was Dr. Sarah Philo. Thanks so much for joining us for our first episode of Absolute Genius. Stay curious, and we'll see you next time. That recording session was a fever dream. Yeah. Us off the rails? Unheard of.
Yeah, unheard of for sure.